Okay, there we are.
Anyway, so we are talking about the actual animals now, because you guys did fabulous on your test. And we are getting into sponges and corals and jellies, and that's what this next test is on. So we start at the most primitive, and we work our way up to the more advanced animals. And hopefully we'll be able to move at a pretty good pace with you and Josie, and we'll be able to move pretty fast through this. So before we get into the actual animals, and we're going to talk about Kingdom Protista, because even though protists aren't really animals, we're just going to really briefly talk about them. So we're definitely going to focus on Animalia because this is a zoology class and they're the ones that are the main focus. We're going to briefly talk about Kingdom Protista. So all of this is important, so you should write all of this down, but we're going to focus like maybe 3, 4 slides on Kingdom Protista. Kingdom Protista is, and this is all domain Eukarya. So Kingdom Protista are single-celled organisms, and when we were looking at pond water under the microscope, most of what we saw under there were protists. They are things like amoeba, euglena, paramecium, mostly aquatic, and they're going to move by a variety of mechanisms. Locomotion, reproduction, and feeding habits are all very primitive, and that's how these things get classified. Now, when we were looking at the pond water, the exception was the dragonfly larva that we found. Obviously, that was a more advanced animal, was not in the Kingdom Protista. But a lot of the other things that we found under there were. I go home because I was shopping with my daughter this weekend.
Did she pick something?
She did. I'll show you after this. Apparently, royal blue is her color. And as soon as she walked out in it, like my jaw dropped. She was like, she's so gorgeous. It was a lot of fun. Even if I was in denial the whole time that she's that way. Even homecoming?
Yeah.
Yay.
She's hot pink. The picture like in the bag for the color reference.
Picture one. Aw. Love to see.
I can see my
Nice.
arm.
So amoeba, here's what amoeba looks like. Very oozy. They want to call it pseudopods. And pseudopods, here's an example of pseudopods. Pseudo means false and pod means foot. So they have these extensions that come out and project outwards. And then basically they like suck up the cytoplasm behind it. And then they will stick out another one. And then they'll suck up all the cytoplasm behind it. So they make these false feet, and that's kind of how they move around, called a pseudopod. And it's a movement of like, sticking out these false feet. And it's also how they eat things. They'll surround things with these false feet, and the food will get kind of stuck in there, and then they just kind of engulf it. So it's really kind of a ooziness for the amoeba.
You know those like fidget toy things where it's like a tube, like the like brightly colored like glycerin.
Oh,
and it
yes.
goes whoosh.
Yeah.
And I hear that I think is like sliding it across the tube.
That's exactly what it's like with them. Yeah. So it's like a pseudopod just like that. So here are some more examples of protists. Slime molds, amoeba, euglena, dinoflagellate, paramecium, diatom, and microalgae. So all of these would be protists. A euglena has a little whippy tail. It's another major type of protist. Euglenas are found in pond water. The other cool thing about a euglena is they have a lot of chloroplasts, so they're able to photosynthesize. So this is an organism, not a plant, that is capable of photosynthesizing. So nucleus, chloroplast, photosynthesize. And then paramecium, another protist, they have kind of cilia outside cilia are the little tiny hairs and surrounds them almost completely. They have a lot of other kind of structures that they kind of have a, not really a mouth, but an area that they kind of engulf their food with. And all these little pores all around them that they eat as well. So, and little vacuoles, food vacuoles that they digest their food with. Alright, so that's really all we're talking about with protists. That's the extent of our protists, because I really want to get into the cool stuff. Now this slide is an important slide. Throughout the rest of the semester, we will keep coming back to this slide. This slide kind of keeps us on track, and we go through each one of these. So it shows us the order that we will be learning stuff and the classification system. So we have domain, kingdom, and phylum, and then once we get to the phylum Chordata, it breaks it down even more. So for the first part, we're going to be learning about the major phylums until we get to phylum Chordata. And then once we get to phylum Chordata, then we start learning about classes. And we'll break it down by class: Agnatha, Anaphistoma, Chondrichthyes, Osteophyes, and so on. So for this first part, we're going to learn about phylums. Phylum Porifera: sponges. Nidarians are like jellyfish and corals. And then Platyaminthes are worms. So we've got flatworms. Nematodes are roundworms, and annelids are segmented worms. Mollusks are shells. So these are like mussels and clams and things like that. Arthropods are insects and crabs, things that have those hard outer shells. And then echinoderms, these are sea stars. And then we get to the chordates, which are the really fancy stuff that have backbones, and then we follow the things that have bones, and these are the more advanced. The fun thing about learning it this way is we look at the evolutionary changes through time, and as we look at the evolutionary changes through time, we'll be able to dissect accordingly, and we'll be able to observe the internal features. So it's kind of like a comparative anatomy class. We will compare the internal structures as we dissect. Our first dissection will be an earthworm, and following that up with a do we do squid? Squids and the mollusks. So we will do a mussel dissection, which is like a clam, and we'll also do a squid dissection that after the worm. And then with the arthropods, we do a grasshopper and crayfish. I did order a sea star, but that's not here yet. I don't think the mussel is here yet either, but I know we do have a squid. So, but anyway, so we will always come back to this slide, and we will always know what we've done, and it's also fun to kind of look back and know what we have gone through as well. So that way we can kind of keep track of where we are going. We will see less birds as time goes on. We'll also see less arthropods as time goes on, because getting into the fall season, the arthropods and the birds are going to start to go away because of migration and because cold weather is going to kill the birds or the arthropods. Alright, so this you do need to write down, because also getting into the actual animal section, we're going to have a responsibility of identification from now on. You will need to know the classes and be responsible for identifying just the classes that are listed on here. So obviously all the domains and kingdoms are going to be the same. We're always going to have domain Eukarya. We're always going to be in kingdom Animalia, but for this one we're starting with phylum Porifera. Porifera is sponges. It's the most primitive animal that we have. And there are 3 classes of sponges that you're responsible for. There's actually many more classes of sponges. But the only one, and even more that are extinct, but the only ones that you're responsible for knowing, that I'm holding you accountable for knowing, is Calcarea, Hexactinilidae, and Demispongidae. So also all of this you should write down. With every grouping of animal, I always try to talk about the species as a whole. And then after I talk about the species as a whole, I'll give specifics on specific animals. There will be pictures as well, and the pictures of the animals I will put in Google Classroom because there will always be some animals you will be responsible for identifying. Now obviously with sponges we won't be able to go out and actually look at sponges in the wild. I think there are, I can't remember if there's any, yeah, there's only a few freshwater, it's right there on the board. Mostly saltwater sponges, and there's only a few freshwater sponges, and I don't think there are any that are around here. But most of them are saltwater. So they're the simplest of all living animals. When the classification system was first made for these guys, they were thought to be plants, and it was, I mean, it makes sense why they thought they were plants. They look like plants. Nobody would believe that they were actually animals, that they were, you know, living animals. They just grow in a coral reef and they look like plants, but no, they are animals. They don't really have true tissues or organs. I mean, if you think back to what, you know, you learned in anatomy, or cells, organs, tissues, organ systems, they don't really have the organization for tissues. Their cells don't work like that, and they don't have organs either. Once they attach to a solid surface, they just kind of stay there, and they filter food from the water. So the water flows into them, and they filter the food out, and then the water comes out of the center. So the water filters in through the sides, and the water filters out through the center. While they do have soft parts, their parts have this kind of scratchiness that is called spicules, and the spicules are actually made of silica. Do you know what silica is?
Is it a mineral, I guess?
It is kind of a mineral.
I feel like I hear it referred to a lot when kind of like volcanic activity.
It's kind of glassy material. So it's really sharp and scratchy like glass. And there are actually about 9,000 different species of sponges. So sponges may not seem too exciting, but the fact that there's 9,000 species is pretty darn impressive. I forgot that we have some examples of sponges in my cabinet of dead things.
Oh no, I forgot I'd be on camera. Hi,
The
Josie.
only one that ever watches it is
And that one guy that left YouTube comments under. Was it a guy? I think it was a guy. Please do medical videos.
Yes. Is it hard?
Yeah, but it's not like scratchy.
Does it smell?
No.
I mean, it is a dead animal. You've seen the YouTube video, no, not YouTube, TikTok, of a person who's like, I use the sponge as toilet paper for my family. Is it real? Or is it like fake? Have you not seen it? You
Did they
TikTok,
take
right?
Yeah, to become slot brushes.
You should look
Ow.
it up. She's like trying to save money. I don't know if she's
Nope. Like the
Amish or
extreme
something.
frugal, but
Yeah.
like zero wasters.
Yeah, but they have a sponge. They keep the sponge. It's on a stick, and they keep it in a bucket of vinegar.
It's giving like the ancient Romans.
Yeah. And they keep it in a bucket of vinegar on the back of the toilet. And I can't tell if it's real or if it's like a joke.
Satire.
Yeah.
I don't think I could do sharing poop stick with my brother. I don't think I'd be able to do that.
You should look it up tonight and tell me.
On the school room. I don't know if I could do that. I'm all about being more earth friendly, but that's I think where I draw.
I think she calls it demispongy.
I can't.
But there's a lot of questions to learn.
I imagine that comment section is probably insane.
But I genuinely can't tell if she's serious or not. I really can't. And I'm like, I just don't know. I just don't know. Cause it's abrasive. It's got the I don't know. I wouldn't do it right
Anyway.
So here are some of the different kinds of sponges. They come in a lot of different colors and a lot of different varieties, and they are typically found in coral reefs. There's a variety of coral reefs, a lot there's some around North America, but most of them are found in tropical regions. Great Barrier Coral Reef is found around Australia, and that's one of the main ones. So sponge cells are specialized to perform different functions. They line the outside of the sponge, they contract slightly to change the shape. There's some that are multi-purpose, and the really important ones are called collar cells. Collar cells have a flagella that actually move water around to help filter it. So the sponges are not just sitting there as a passive animal. They have these collar cells that flick their cell, like the flagella, to actually move the water through. And we're gonna watch a video, that'll be your next assignment, is to watch these videos. And the video, what they do is so neat. They put this like neon dye on the outside of the sponge, and then you'll see the sponge actually suck up the dye and shoot it out the center. And it's the neatest thing because it shows that the sponge is actually actively sucking up water. Because if it wasn't, the water would just be like passively moving through, but it sucks up the dye so fast and it like shoots it out like a little mini volcano. It's the coolest thing. And the way the video demonstrates it makes it so obvious that the sponge is doing it intentionally. And the dye doesn't hurt the sponge, like it's non-toxic.
How did they figure out that they're not just little plants in there? They're specific.
No. That's a good question, but I'm not sure. Somebody had to have taken it under a microscope and then seen the different types of cells and started putting it together. Well, first of all, there's no cell wall, and plants have cell walls, and there's no chloroplast. And
Just imagine the guy that like figured it out. His friends were like, "Why do you care?"
it's just a sponge. Well, but there's no cell wall. It's not green. It's not a plant. So here's kind of how the process works. It's got all of these little openings on the outside, and these are pores, but these collar cells are have these little flagellas that are bringing the water in and kind of sucking it and drawing it in. The spicules are represented by these little blue pieces, and the little blue pieces give the sponge its shape and make it kind of abrasive. This center is called the osculum, and that's where the water exits. So it sucks up the water through the sides. And that's going to actually filter the water too, because that's how the sponge eats. All these particles and things that are in the water are going to get filtered out as the water is sucked in and trapped, and the sponge is going to digest that. And then anything that is left over gets put through the center and out through this osculum. There are different kinds of cells. Amoebocytes, anything with the word cyte as a cell. Amoeba means that it moves like the amoeba we were just talking about. There's mesenchyme, which is the middle kind of, it's like jelly-like fluid here in the middle. It's not really a cell, it's kind of a fluidy stuff. Almost like a cytoplasm, but not quite a cytoplasm because it's not really in a cell, it's kind of in between. And then in ostium, which is the little pores in between. So here's another view. What's interesting about them, we haven't really gotten to that yet, but we will, is that they do reproduce by budding. So they can break off, and this little piece can break off and start a new sponge somewhere else. So we have amoebocytes. We also have these things called pinacocytes, which are the cells on the outside, the epidermal cells. And spongiocele, which is the inner portion, like this inner section here is the spongiocele. Ostium, osculum is the opening. And ostium is the pore that goes in. Porocyte is the pore cell, so the big pore cells. Here's a porocyte here, here's a porocyte there. And then the mesophyll is kind of the inside. Here's what the spicules look like up close. You can see why it's so scratchy and very pointy.
Not doing this sponge toilet paper thing.
You can also imagine, because of these spicules, it's very difficult for many animals to eat. Now there are some things that definitely can eat sponges. Sea stars can eat sponges, but also sea turtles will eat sponges.
What's the nutritional value there?
Probably not a lot, especially because you have to digest this.
Yeah.
So not a lot of things do like to eat the sponges, but there are some things that do. So there are three main types of cells: the pinacocyte, the choanocyte, and the amoebocytes. The pinacocytes are on the outermost surface of the sponge, and they're like the protective kind of outer portion. The choanocytes are the collar cells, and they're the inner surface, and they're the ones that have the flagellum to move the water. Amoebocytes, they're multi-purpose, and they can kind of fill in the gaps. So they're kind of like stem cells for us, the stem cells that can become anything. That's what the amoebocytes do. They can become a choanocyte, they can become a pinacocyte. They can also, we'll learn later, become a sperm or an egg cell later on. But more often than not, they fill in the gaps and become a choanocyte or pinacocyte. A lot of times sponges are just going to reproduce by asexual reproduction. They don't typically become sperm or egg cells. Mostly it's going to be asexual reproduction. So here's just another way to look at it. A spongiocele is the inside. Cele means cavity. So the spongiocele is this inside cavity. And then ostia is the holes. Mesophyll is the middle. Think meso from middle. Osculum is the opening. Close up of it, we have the oocyte, which is an egg cell. Amoebocyte can become an oocyte. This is a sclerocyte, which makes the spicules. Here's our choanocyte, is the pink thing with little wiggly tails. This flat one is the pinacocyte, which makes the outer portion. But see, there's really not a tissue. It's like, 3 layers here, and there's stuff kind of in this mesophyll, which is in the middle. But other than that, it's very, very simplistic. There's really not a whole lot going on. There's just, a handful of cells between them. So as for reproduction, most sponges are both male and female, so they have both. And the collar cells and amoebocytes change into sperm or egg. So most likely you're going to get a sperm cell from a collar cell because they already have the flagellum tail. And the amoebocytes are going to turn into an egg cell. So they're going to reproduce outside by just releasing into the water. In fact, a lot of oceanic animals do that. They just release the sperm and the egg into the water because, hey, the water mixes it for us. It's just perfect. So that's what a lot of them do. Like, it's called spawning. The corals will spawn, the sponges will spawn, and oftentimes they coordinate it to all spawn together. It's a higher chance of survival that way. We all spawn at the same time, less chance that my sperm and eggs are going to be eaten. But they can also reproduce by budding, and typically what they do is they will reproduce sexually when the water is warm, and they'll reproduce asexually when the water is cold.
I can't stop yawning.
She needs to stop eating. Can't. So here's kind of how it works. This is a bud. It breaks off, and then that little piece will float away in the water, land, and become a new little sponge. So any part can break off and create a new little sponge. But you can also have the egg and the sperm form. Typically, again, it's a cholanocyte or collar cell that will turn into a sperm, and then an amoebocyte that will turn into an egg. They'll release into the water, and the egg and the sperm will join each other and create a new little sponge, and that is sexual reproduction. So a sponge can do either one or both, depending on the sponge. And the idea here is, again, to reproduce away from the original sponge so that it has more space. So when we get to these pictures that look like this, that have the scientific names on them, these are the ones that you're responsible for knowing for the test. These pictures are in Google Classroom and student notes. And the way I do this is I will have a variety of these pictures on the test. Typically, I give you multiple choice with these. So there'll be a drop-down menu, and you will have words to select from. So you don't need to memorize the scientific names, but you will need to be able to tell me that this is a giant barrel sponge and be able to identify it. So this one is considered a giant barrel sponge. They can get quite large. There's a fish down here, kind of for reference size. Obviously, it's really hard to reference this if there's not a person or anything standing nearby. This one is a red finger sponge. Obvious why that one's named. See, I like these names. These names make sense. Giant barrel sponge. That makes sense to me. What?
You said this is pretty reasonable.
I know. Sometimes names are not. Red finger sponge. That one also makes sense to me. Yellow tube sponge. Again, somebody thought of it. A good name. That is a good name right there. A yellow tube sponge. This one, I don't know. What's a row-pour rope sponge?
Tongue twister.
I know. Why? Row-pour rope sponge. I don't know why. But it is kind of odd. I mean, look how long it goes.
It's like twisty.
Yeah.
Swirly.
All long and kind of laid out, and then at the end it goes whoop.
I wonder if maybe that, do you think that would be influenced by like current?
That makes a good point. I never thought of
It's
that.
just easier to grow in that direction. Like,
I'm sure that's probably why. If we looked at which way the current was going, I'm sure that's a big portion, a big reason why it's growing like that.
or they stick around in places with stronger.
That's because other ones can't. Probably my guess. Well, and in a place with big current, that would push the water through them so they could eat better. Because
If
they,
it's too strong, it would tear them off.
yeah. Because they're stationary, the water has to go through them so they can eat. That's a good point. probably right. Okay, so those are the different types of sponges. We've got a giant barrel, red finger sponge, yellow tooth sponge, and a rope or rope. If it is not in pictures, and I'm not going to put it on the test because that's not fair of you to know any pictures that I have not gone over with you. But anything we've talked about in the slides and anything that's in student notes is fair game for the test. Alright, so that's all the notes we're taking for today. Tomorrow we will go over some more. But I will give you your assignment, and it's to watch a video about sponges and looking at the unique characteristics of sponges and how they're related to the coral reefs. So, let's go on with that. As soon as I push pause.